So I'm going to try and answer this question, which is, of course, a debatable, a hot, controversial question, but indirectly. So we all find the evolution of the universe fascinating. This is a simulation of it in Heidelberg. What I, as a biologist, find incredibly fascinating is how organic life emerged from this entire process. And not just organic life, but how we evolved out of this process. During this process, I think that what we tend to overlook is that at every single cell, as it divides, it is becoming connected to the world in some way. It's not in isolation, it's not in a vacuum. Every molecule that is evolving is becoming connected to the world. Every single cell and every single molecule connected in some way to the world. It's a double bridge, it's a two-way street. The world can talk, speak, communicate to every single cell and every single molecule. There is no molecule that is in a vacuum. How many are there? Well, 37 trillion cells in your body, approximately. Between 2 and 5 trillion molecules in every single cell. 70% is water, 1% are inorganic ions, inorganic compounds, the other 29% or so organic compounds. This is an incredible number of molecules connected to the world. So this is a yotta. You are a yotta machine, a yotta, 10 to the 24 molecules. You're a puppet of the universe. Your whole body is coupled with strings to every single facet of the universe. We can't even see all the things we're connected to. Every single molecule. There's where something could happen, affect your movement, affect that cell, affect that particular molecule, and back. So you are a yotta machine. This is very important because this is where I personally believe the real intelligence of biology comes from. It is not in solving an equation. It is not in doing a computation. It is just in a molecule jumping to the left, jumping to the right. And that is why we can do such incredible computations using the power of a light bulb, not the power of a nuclear power station to drive a supercomputer. That is where our computation, that is where intelligence is coming from. And if we want computers to beat humans, we have to beat that. That's the benchmark. So I don't think that's going to happen anytime soon, but we're going to try. So if we look at the human brain, it has evolved from mouse to man, from mouse to human. The brain has expanded by 3,000 times, and about 1,000 times more neurons, double the number of synapses from the mouse. It's not just in the numbers. It's not just that we have more of neurons or computing devices. It is that they are more organized. They're more structured. And this structure somehow is, and that's the somehow that we're going to get towards the end of this meeting, this structure somehow is leading, in Christoph and Giorgio Tononi and others' words, leading to a causal mass of interactions, a critical mass of interactions. It's like a critical mass in a nuclear explosion that allows the evolution of consciousness, which allows us to perceive the world. And for me, the single most fascinating question is how the universe evolved this brain to become conscious of the universe. Imagine if there was no brain. Who would know there was a universe? Nobody. You would have no pain, you would have no color. Nobody would know there was a universe. The brain knows that there's a universe. But how? How does it do it? How does the brain evolve this structure, this machine that suddenly can look up at the stars and say, what are you? What am I? Where did I come from? Well, the approach that we're taking is to build it and step inside of it and see how it does it. But not just see the electrical activity and the activity of every molecule and the activity of every synapse and of every neuron, but actually ultimately see how it generates back the perception. Can you put this on? To the perception. And that is when we will know we've understood the brain. Now, this endeavor to build the brain or construct it, reconstruct it, is not my idea. It's not a new idea. It's been going on for 100 years. It was started by this amazing guy. He was a physician, and he sat instead under a gave up looking after patients and started staring under the microscope and drawing all the neurons. He basically drew almost every neuron that there is in the brain. It's a fantastic, lifelong mission. He did it with a pencil, but today we can do it with computers. So we are basically doing what he did, but trying to build them in a digital form. Now, when you want to build the brain, of course, very aware of all the controversy that it generates when one says one wants to do that. One of the real reasons, one of the real justifications for the controversy is people say, but you know, we don't know enough about the brain. How are you crazy? How can I come build it if you don't even know enough about the brain? Do we have enough data? So you have to ask, well, should we wait for the data? Should we wait until we have enough data and then we start building the brain? Well, then you have to also ask, can you get enough data? Can you? So if you think we should wait, can you go and get it? Can we experimentally map the brain? Is it like the human genome that's got 3 billion base pairs and we can just go, and there it is? Well, it'll probably take me an hour to go through all of this, but basically 20,000 genes, every cell expressing about 5,000 to 10,000, between 2 and 5 trillion molecules, about 1 trillion organic molecules, 1 billion major proteins, 450 ion channels, over 500 different receptors with many variants, 737 brain regions according to the Allen Institute, 5 major cell types on average per brain, 3,000 major cell types, 70 million neurons to 86 billion neurons, 1,500 kilometers and 3 million kilometers of fibers in your brain. 600 different brain disorders, approximately, that we have. And then you have over 5,000 remaining species. Individual variations, we're all different. Which brain should we map? nature versus nurture. Your genes are going to slightly change things. The environment is going to slightly change things. Where do you move to? Which one is going to map this one? Is that representative? Spectrum of cognitive which ones do you derive? We need to get the neural correlates, but of which ones? Of all of them. Many, for each animal, for each species, for each disease. Experimental variations. The biggest lesson we've learned in the Blue Brain project so far is that there is no experimental ground truth. There is no experimental ground truth. The simplest data set that there exists, that you can think of for the brain to measure, is the number of cells in a particular part of the brain. Our analysis shows that over thousands of studies that have been done on measuring how many cells there are in any part of the brain, the experimental variation between laboratories is threefold. Threefold. The long tail is 13-fold. Which lab are you going to believe? We've seen that there's a high experimental variations when you look at differences in morphologies of neurons than differences across species. Which data are you going to believe? So I think that if we believe we should wait until we can record and measure every single data point that we need to blindly build the brain, we're going to wait for a very, very long time. Fortunately, that's not a scientific approach. It's not scientific to say that you should measure everything, because we know from Claude Shannon that you only need to do that in a random system, and the brain is not a random system. So if you want to build the brain blindly, you need all the data and basically virtually no knowledge. If you get more knowledge, you need less data. So the real question we should be asking is, what's the least amount of data I need to build the brain? That is the pressure. That's the evolutionary pressure we should be putting onto our science. Exploit the relationships, the interdependencies, and be able to infer as much as possible so you can fill all the gaps of knowledge, test them, validate them, and move on and predict more and more. The one way you can do that is to use the data and test it by building a hypothesis. And the hypothesis is a reconstruction where you can use the data to build these models and test your knowledge. Falsify it, go back, and keep the cycle of science going until you've accounted for all the data and you have mastered and accumulated the set of knowledge that's going to do it. This is an example. You could say, how do I measure all the synapses? You have 1,000 trillion synapses in your brain. Are we going to measure them all experimentally? Yes, we need a lot of experimental data, okay? So don't get me wrong. You need tons. We should never stop. But how are you going to get all of them for almost any condition? Well, you have to get an algorithm. This is one algorithm we developed. 4 basic principles that we found. 4 basic rules of how neurons connect can be used to follow the branches, decide when they touch each other, and based on that, decide whether that's a biologically viable place where a synapse can form. Algorithm, you can derive, and you can now obtain all the synapses in this little piece of the brain, which is about the size of a pinhead, 8 million connections, 36 million intrinsic synapses. We can infer that this, from the space remaining, 140 million other synapses. The rest of the brain is talking to this pinhead through 140 million other synapses. By looking at the morphologies, if you go out into the tree, if you're a botanist, you look at the tree, you may look at trees many, many times and just keep drawing them, or you can just study their morphologies, find a mathematical formula, and grow synthetic trees. Well, you can do that with neurons today. You can grow almost any neuron. We have an approach where you can now grow all the different dendritic branches. We're moving to sort of almost an AI-based way where you can take your phone and scan drawings of neurons from papers and generate 3D models. Of course, you can validate them, you can combine them with other data and so on to get them accurate. Together with Idan and his amazing students, we developed now an algorithm that allows you to automatically model and capture very detailed biology of a single neuron that replicates how the neuron behaves And you can do that for millions, if not billions of neurons. Another area where you can gain and you don't have to necessarily measure everything is by looking at some of the data across species. And this is a good example. Here we actually clone and sequence and use biophysical screening to measure the biophysics of all of this family of ion channels. You have many different voltage-gated channels. There are potassium channels. They basically stop a cell from when it's depolarized or excited. They reduce the excitation. So we had to map them. And when we did that, we looked at them in rat, mouse, and human. And what you can see here is that the kinetics are identical. Identical. So related to this conference, it actually tells you one other thing, and it's going in the same direction, the same trend that we've seen. The basic building blocks from mouse to humans has not changed. What changes is the regulatory sites. And in fact, you'd be surprised to actually see a change here, because if you look at the gene sequence, they're identical too. So where the differences are in the regulatory sites, how you control your building blocks, all the hooks that you have to control the building blocks. But again, this allows you now to transfer knowledge across species and help you find the data. So there are many approaches. Here we can now build algorithms that draw in the data. This is taking all the atlases of the Allen Institute, actually computationally analyzing how many cells there are, where they are, how they're located. And that allows us now to build very detailed atlases, cell atlases, not drawings or pictures or stains, but atlases that actually tell you how many cells there are. This is for both neurons and glia, and you can account for basically every single cell in the mouse brain, 74 million cells. You can look at the projections and find rules. This is one rule where we're working with the Wuhan, and also it's a collaboration with the Allen, trying to identify what we call projection rules, or p-types of cells. And based on these rules, and combined with lots of other data, you can actually identify using DTI in this case, but we have many different approaches, neural seeds, and then you can make a prediction of what is the projections. Here we're using the Allen Institute has about 3,000 experiments where they injected dyes in different places, and you can use it and calibrate and validate the model that it will have the same projections. So understanding how the entire mouse brain is connected with its 1,500 kilometers of fibers is not an unsolvable problem. You can computationally now synthesize all these morphologies so that you have neurons to be able to put into them. And here we use that to actually build one complete microcircuit with all its different, what we know of all its different cell types that there are. You can also now computationally synthesize with a KAUST in Saudi Arabia. We have a collaboration where we take electron microscopic scans of glia, this is astrocytes, protoplasmic astrocytes, and we can computationally synthesize them. So now you can make beautiful synthesis where you can recreate the end feet onto vasculature, because they control how the blood supply comes to the brain. They communicate with the neurons to support metabolism, clean up the potassium, help the supply of neurotransmitters, and so on. And you can extract principles to allow you to, these are all computationally synthesized, to produce tiling so that you can account for and describe all the glial cells in the cell. And there are 3,000 here, so it's 10% of the neurons are protoplasmic astrocytes, which has been derived by combining all of this data. You can now start adding them to the blood vessels and create a blood pumping digital microcircuit. One can go further, and here we're building an atlas of basically every metabolite that is involved. There's over 5,000 metabolites involved in this neurovascular glial system. There are compartments. Every compartment is involving about 1,000 interactions. So we're building a complete map of that, and as you build that map, you can start modeling that map to understand how it works. Because you have compartments, you need to know where these proteins go, where these molecules go. We're also building these subcellular generators that produce the endoplasmic reticula, the Golgi apparatus. Of course, your first draft that you make is not anywhere near correct, and that is why we go through this cycle where we build, it's a hypothesis, you test it against the data, you see if any discrepancies, you go back, you get more data, you revise your knowledge, and you keep the cycle going until your knowledge goes up and your data goes down. That's the target. These models today are becoming very precise, and they will become more and more EM precise. This is some of the models that we can build today. This is some of the simulations. So, simulation of a slice. When I started in Menachem Segel's lab in the Weizmann, I did slice recordings. I cut a piece of the brain, I put it under the microscope, and I did these experiments. It was an amazing time. And so we recreated a virtual slice. You can do experiments on these. You can do experiments that copy papers that have been done on real pieces of tissue. And so far, without touching this reconstruction, we've been able to recreate and basically validate it against a vast number of different experiments. You can go further. You can isolate the somatosensory cortex, going from a microcircuit to a region with this atlas. Basically, we're building a click-build strategy where you build all this knowledge into algorithms. So you go to the thing, you select somatosensory cortex, you can see all the neuron numbers, you can see where they are, which layers they are, and then you can basically load in your neurons, connect them with the algorithm, and run a simulation. This is a simulation of somatosensory cortex, which we completed last year and are still in the process of running through the cycle, testing it against other experiments, and making predictions of things that you cannot do experimentally. And then you can go further to, this is the neocortex, you can isolate the neocortex, but here there's all these curved spaces, so it's a new computational challenge. You need to know which is the orientation of the neuron. So you could select orientation. We used a lot of data to decide which is the orientations of cells, so we have an atlas with orientation, so you can load in the neurons, or you can computationally synthesize them in the correct directions. And then once you have that, you can start loading in your neurons, connecting them, and simulating them. You have your neurons modeled before, your synapses, you apply the data-driven approach to define how the synapses are working. Can now apply the same approach that's been developed for the neocortex, you can apply to other areas. Here we did it. It took 10 years to do that small little pin drop. It took a few months to do the whole of the somatosensory cortex, 9 months in fact. It's getting faster and faster and more general. Here for the Human Brain Project, we've been doing this as a community project, lots of people involved. But within a very short period of time, we could actually create a first draft of the hippocampus. And this is a hippocampal simulation where you can now start recreating and doing in silico or virtual experiments similar to, looking for place cells or looking for theta oscillations, and you're doing learning experiments and starting to ask questions about where do memories lie, for example. We're taking this much further now to reconstruct even other areas. This is the dopaminergic system, and so on. The main point is, this is happening, it's happening faster and faster, and it's happening more and more accurately. The first skeleton draft we have of the whole mouse, 74 million neurons. It's not all the synapses, but it's 259 billion synapses. And it provides us a framework to start running the cycle and improving the accuracy of these models until they capture all the data that we have and account for the observations that have been seen. The one thing you'll notice in this is that you see with your whole brain. You do not see just with your visual cortex. You do not just smell with your olfactory system. The whole brain is involved, and it'll come back to a message, I think, later. Of course, you can take it further and start building a little virtual mouse and coupling the brain to the body and start to see whether you can do in silico experiments, behavioral experiments. You can select a behavioral task. I think that this figure is from the Allen Institute, but I'm not sure. And you could say, oh, I want to replicate this experiment. So there's also in the Human Brain Project a collaboration to rapidly build oneself a little mouse, build the muscles, the spinal cord, connect the fibers, build a brain model onto it, and to start to test whether this model, whether it's a simple model, a more complex model, more realistic model, can execute the behavior. The power of a detailed model is that when it can replicate the behavior, you'll be able to say which neuron was involved, synapse was involved, molecule was involved, and so on. And our goal, of course, is to be able to deploy these into all kinds of environments where it can do all kinds of artificial and useful things, such as understanding how the body moves. I'll just share one example, and it's relevant to this particular conference. One example of an insight that we made. So one thing that was known for a long time, but we could see it very clearly, is that the brain lives on a spectrum of states, going from regenerative state, which is a highly synchronous state, to an asynchronous state or non-regenerative state. And it lives along the spectrum. And in fact, there is one parameter that can shift it along this state, but we know now which molecules shifted in this state, we know which synapses shifted in the state, but there's one parameter that shifts in the state, and this connects with Mike and Nedergaard's lecture that we heard yesterday. You add a bit of calcium and it goes to sleep, you remove the calcium and it wakes up. And this is important because we identified a very sharp transition, and it's only at this transition that the Science and Nature papers that are published findings on in vivo, the whole brain, about choristers and certain triplet behavior, very interesting behaviors of neurons. Those phenomena, they occur at this point, at this transition. It's like a cliff where if you go just a bit further, the brain runs away with itself. That's a critical point where very interesting phenomena that have been described before are occurring. But what is very important is this calcium level, and I'll tell you why. What we did is we built many different versions of models from different animal data. It's like building different individuals or customizing the model to different individuals. Different sizes of the layers, numbers of neurons, types of neurons. So we could build different types of microcircuits. And what we discovered is, because we wanted to know what accounts for individual variation, what we discovered is that each of these circuits actually reacted quite differently, like normal animals do. They had a biological variability. But when we looked at the spectrum, we discovered the cliff position was very different. But then we just added and removed a little bit of calcium, and they all became the same. We could normalize individual differences. So with a bit of calcium, you could all become the same. Beautiful, different, diverse, amazing butterflies all become the same. So who are we if we can all become the same? But what it does tell you is that you are not your connectome, you are not your neurons, you are not your composition, you are your state. You are the state that you can move yourself into. And Felix is getting nervous, he's German, he's very precise, he never lets me get away with anything. So he's not going to let me get away with talking any longer, so I'm not going to tell you how long it's going to take for all of this to happen.
I'll give you that minute.
Except to tell you there's one thing, that to build a computer, just physically to build a computer that is going to be able to simulate the human brain with 1,000 trillion synapses for each neuron, with 90 billion glial cells, 450 billion vascular end feet, supporting 450 trillion synapses, 1 trillion molecules per cell, 1,000 reactions per molecule per second, 20 diffusion jumps per second, 10,000 time steps. To get this right, you have to simulate about every 25 microseconds. You need a machine that is about 10 to the 30 calculations per second. Okay, so we run out of names because they stop at iota. But, so you have to wait a bit longer, but you don't have to wait that long. It's going to happen probably in the next century. So in the 22nd century, that's going to be possible in real time. The human brain in real time in the 22nd century. But you don't have to do it with all that level of detail. You can make it simpler. You can reduce the number of neurons. Let me just go back. You can reduce the number of neurons. You can do low resolution. You can group the molecules. You can also just throw away the molecules and make phenomenological equations. So you can get closer. You can go all the way down to point neurons. So point neuron model of the human brain is feasible already, basically within the next year or two. But what we need is we need to know how many types, how are they connected, very much like I showed you for the mouse. The last thing I want to say is that when we won the Human Brain Project, and since we are in Israel, the most important quote we got was from Shimon Peres, and he said, the reason why this is absolutely important is because we are strangers to ourselves. So one of my dreams is, and Idan, we started this very early, is that we would love to take the whole planet on a voyage through the brain so that we can see how it is reacting to the world, and hopefully in the future we will have the EM squared equation that will translate that activity into a perception, which will tell us that that is when we can understand the brain. That is the Blue Brain team. Felix is the co-director. Without him, none of what I spoke about would have been possible. Thank you very much.